developed the first PC-based GIS for insect forecasting in the world. And along the way, counted probably 100 different species of insects around the world, and especially in Alberta. And now I teach data analysis at the university, as well as the distribution of organisms. So I have an IPM perspective in my mind all the time when I talk about insects and crops. And I think most of you know all about it, but just to be sure, I want to emphasize that it's based on action thresholds, which are themselves based on economic injury levels, which you can't do without pest taxonomy. And monitoring is where we're going to focus a bit today, maybe on a few things you haven't thought of. And of course, the other side to IPM is prevention and biorational control, so that it's environmentally sustainable and doesn't interfere with natural enemies and fits into a proper economic and environmental risk management scheme and so on. But today we'll talk mostly about numbers. Okay, so a couple days ago I was out. I saw my first wheat stem sawfly of the year. So immediately, what do I think? How many more are there of them? Where are they distributed? Are they along the edge of the field only? Did they come from this field or did they fly? Because obviously this one is capable of that. So I switched gears a bit and spent some time on Highway 61 and surrounds, searching out to see if I could find a hot spot of wheat stem sawfly. Fortunately, thanks to breeding and so on, this is not the problem it once was, but it's still out there. Now, what are these things that we use when we do counts? Why count at all? Why not just say they're bad or they're not bad? Well, we can actually calculate an economic injury level. And it used to be that was only based on crop protection cost. Now it can be based in part on environment. Sometimes we would like to avoid damaging the environment, and sometimes we would like it to work for us in pest control. So the lowest number of insects or other attribute of the insects that we're measuring, like activity, that will cause a yield loss that would balance the control cost. I think everybody has a good feel for that. And the action threshold, or the economic threshold, is the pest density at which a management action prevents it from getting any worse or continuing to cause damage. There's a problem though, of course, and that is these are not single numbers, they're ranges. And we can only talk about this problem with respect to both precision and accuracy, and that's why I put them in the title. So the economic injury level is plus or minus what, and under what conditions, and how warm, and what part of the year, and how will the crop respond, all these questions. And in the economic threshold, well, if I go out and say, I'm going to find 3 in 10 sweeps with a net, and then I'll take action, and I find 1, 7, 2, 0, 12, what do I have? How much of that field is actually at the economic threshold? Should it be sprayed or not? Should I come back again or not? This is the question. So when we look at sampling, it's got to be efficient, it's got to be cost effective. We can't fool around and waste time, but we want the precise information. Well, what's the difference between precision and accuracy? They are based on variances, which is the variability of the data, the means, which are the true value that's out there that we're trying to estimate with our sample mean, and the distribution, is it spread high, spread low, or mostly in the middle. So you probably know all about this. If you've ever had to practice until you get something better, like target shooting, or maybe adjust a sight. But precision is low in this particular example, but accuracy is high. If you looked at the example, the average of all the points in this example, it's pretty well in the center of the target. So that's accurate, not precise. This one's very precise, but it's not accurate. High precision without accuracy is still a good thing. All you have to do is shift those sights, and then you're shooting quite well, right? So that's the thing we have to think of all the time with insect counts. We don't want to spend a lot of time doing a lot of counts, but we can do just a few for a good reason. And we need to take the next step in the interpretation. As it says here, there's two types of people: those who can extrapolate from incomplete data, and the other kind. Here's a quick example first: cabbage seed pod weevil. People know about that, I think. It's been around since 1995. I first started working on it in 1986. We want to sample a number of adults in the field. I took this photo of them clinging to a flower before they lay the eggs. They lay the eggs into the pod in late June. The adults are out very beginning of the year, flying around looking for canola or what have you. We want to count them. So what do? Well, first thing you should probably do, and I'm not going to give you all the threshold values. I mean, you can look them up. They change, right? First thing you should do is consult the government websites and the industry websites. And you could go to this one, for example, and find out a survey map on where they're going to be high and where they're low. This year, they're rather high in some parts of southern Alberta, low in central and farther north. And there's all sorts of information online on life cycle, which is absolutely required, of course, before you can even interpret what the counts mean. But here's an example of how you might count. You wouldn't just step into a field and count, because then what you have is an estimate on the edge. That's it. The researchers like Thipa here and Hector, and I work with them too, go out into the field a specified distance, in this case 50 meters, 10 sweeps, 10 sweeps, 10 sweeps, 10 sweeps. And if you find no seed pod weevil in all of those, you have a pretty good idea that there's very few or none. But if you find high numbers and it's repeatable, then obviously you have more confidence in the problem. And that's where the problem lies, that we have to take a look at for precision and accuracy. Now, so accuracy, of course, is how close to what is actually there, which we will never know, but we try to estimate it with our sample. And precision is how wide it is, how variable. And when I say confident, I don't mean I'm doing a good job, but I mean the confidence. For example, if I take 5 measures, take their average, the spread of those 5 measures will tell me how confident I am that the true value falls within a particular bound around that central mean. And if I've got numbers like, 11 weevils, 9 weevils, 10 weevils, 11 weevils, I have very good confidence that I'm in a certain zone. If I've got 0, 0, 95, 13, it's really difficult to say, and probably I have a distributed population and it needs something called stratified sampling, where you would divide up the field and maybe hit 4 corners. Okay, here's another example. Potato psyllid, it's also called tomato potato psyllid, arrived in Alberta at an unknown date in the past. We started sampling for the first time in 2013. We had a national sampling program. I was coordinator and PI on the research program, looking for this insect, which is very common in Idaho, Washington, places like that. Lays eggs on a tomato or a potato. There's the eggs, easy to find. They hatch into immature stages that then form this flying stage, which is somewhat related to aphids and about that size. So we put out thousands across the country, thousands of yellow sticky traps, and then use microscopes to examine them. But we didn't put just one trap in the field, of course, we had to put more, so we have repeatability and some measure of detection. In the end, we would maybe use 4 traps per field. Under research, it was 8 per field, perhaps. Examine the leaves, use a net, use vacuums. This is a grad student, Summer Shah, collecting one of those cards. You can see how big they are in the field. We swept the fields as well. A lot of backup, a lot of time spent examining. 100 leaves are examined looking for the stages because we really thought they were going to show up. So that was an unusual case in which we had near 0 numbers of a very significant pest. This pest, its name is Bacteria, it's a group of psyllids that attack citrus or potatoes or tomatoes or other things around the world, and they carry bacteria. So it actually has a bacterium that it carries that causes zebra chip in potatoes. Very important to catch even the first few. So it's a little different case. Now, why does range matter? I just want to talk about numbers here for a minute. I don't want to get started on it too far. I could talk on it all day, but it does matter a great deal. Why is that? Well, if you take even random numbers, if you do insect counts, that's the same basically. You could do that, but you could even just take random numbers, calculate the average. Now you could assume that's the average for the whole field, or you could continue sampling and calculating an average and an average and an average and an average. What do those averages look like? Well, they end up looking like this every time. And you could prove that to yourself even with random numbers out of a phone book. They're mainly in the middle, and then there's some low and some high. This is why range matters. The variability of that spread is what matters, because if we have an economic threshold that's right in the middle and we hit right on it, half that field, according to our sampling, is below it and half is well above. So what do? Well, we could wait. We could sample a little more intently, or perhaps use the stratified method I talked about, because I think everybody knows that these insects tend to clump in certain parts of the field. Now, maybe I could even wonder if I could share Excel and just show a quick calculation and show that this is not rocket science. Jamie, is my blank Excel showing right now?
Yes.
Yeah, okay. So let's just say I won't spend a lot of time on this, because I know it's boring to look at someone else type a spreadsheet, but everybody here works spreadsheets because they all make a lot of money or lose a lot of money, either way. But let's just do it quickly, and you'll see it's really very simple. And if you had to give somebody an idea of the mean, the average value, and the spread, especially if you're talking to a rather sophisticated agricultural advisor who'd like to know, it's a simple thing. If I found 4, 1, 6, and 2, and maybe 5, so what do I have there? Am I over 4? Am I over 3, or what? Well, I'll just make an average here, do it the quick way. Oops. The average of these is 3.6, so it might look like I'm just barely over the threshold and I should spray the whole field. Well, is that really the case or not? Maybe let's take a look so that we can calculate the deviations. So the deviation for this one would be just the difference between 4 and 3.6. The deviations for the other ones I could just calculate down all the way, and some are above 3.6 and some are below. And of course they would sum to 0 because they're on both sides of the mean. Now, for a long time in agriculture, we've computed a quick and dirty measure based on the squared deviations so that they don't add to 0, right? So I could just say this times itself and then drag those down. Those are all squared deviations. And I now have a value, I have a value here that's the sum of squared deviations, 17.2. So what can you do with that? Well, the sum of squared deviations divided by n minus 1 is the variance. So if somebody said, well, you've got an average of 3.6, what's the variance? Well, you can easily calculate it with a spreadsheet like that. I'd be happy to send this to anybody who wanted to. And another measure, the square root of that is the standard deviation. And that divided by the square root of however many samples you took is the standard error. So now you could actually use these numbers to calculate, well, how much of that field, based on my sampling, if it's good enough, do I feel is below the economic threshold, and how much of it is above? How sure am I that it's really 3.6? Maybe I'm in the ballpark between 2 and 4 or something. So it's these statistics. I'll stop this and go back to insect pictures. The statistics are actually fairly easy to compute, as long as you use proper sampling techniques. So maybe let's just look at a couple of those. I'll maybe share a video here. Let's see if it works. Jamie, does that show for you?
Yes.
Let's just see if it doesn't show how easy it is to use these two different kinds of nets. Oh, it's not very loud. You can't hear that sound very well, right?
No. Or
Okay,
when you
what if
select the screen share, there's a toggle that says something like share computer audio. Just make sure that's flipped on.
You already told me about that, and I forgot. So when I share that, it says, oh yeah, include, there it is, yeah. Okay, now here we go. Stop share, share, and it's gone. I guess I, I guess I should actually have another. Okay, I'll just explain what's happening here. There's two nets. A heavy canvas net is used for cabbage seed pod, weevil, grasshoppers, things like that. A light net for lighter crops, like maybe the top of flax or something like that. If you use a net like that, you should strap some nails or something on the bottom so that you can actually use it and turn it. That thing is 38 centimeters across. If you have a bigger or smaller one, you have to kind of do some adjustments in your head. But you can buy these from UFA and they're standard. Try to go 180. You see I'm kind of swinging past because I want a full 180 there. And then the insects are in the bottom. You can take a look and do a count. At this point, if you wanted to take a photo and send it to an expert, it'd be easy to do. So there, I'll stop that share. Go back to my, sorry, Jamie, is the PowerPoint showing again?
Yes.
Okay, good.
Just need to put it, yep.
I'll stop that movie. Okay, I'm more of a Zoom user. What I wanted to show anyway is that basically you have a spread around any estimate you do, and if that is wide or narrow, it's important to try to get some sense of it. And one quick and easy way, if you don't want to do calculations, if you really like calculations, email me. I'll give you all the Excel files you could ever want. But if you don't want to do the calculation, just take the range, and that is a nice estimate of the variability. And if you really wanted some standard deviation, if somebody was impressing us with their knowledge of that word and asked for it, you just take the range, divide by 4. It's very close. Okay, now a little bit of lore and also some recent research information on what's up with current pests is always listed online in the Prairie Pest Monitoring group, in the provincial agriculture websites and so on. Tyler Wist gave me this one called Field Heroes. It's about the natural enemies. Hugh Phillip, who I knew well, is retired now, and a group made this field crop and forage pest identification guide, which you can find online. There's a ligus bug. And again,
Dan, I think you closed your PowerPoint if you were still showing us stuff. We don't see it anymore. I think you closed that instead of the video.
Okay, thanks for telling me. I'll reshare that. Is it showing now?
Yep.
Okay, there's the Field Heroes banner show.
Yep.
Right, so if you go to fieldheroes.ca or the prairiepestmonitoring.ca, you can find out the latest on not only the pests and their distributions and abundance that year, but projections and the natural enemies. So this is the identification guide I mentioned. Hugh Phillip and a group of entomologists made. And this is a screenshot of the Prairie Pest Monitoring Network that tries to give some latest information on a long list of pests: Bertha armyworm, pea leaf weevil. They have extensive wheat midge surveys now. And some of it has to be taken with a grain of salt, like forecasting species based on air temperature doesn't always work and so on, but it's a very good starting point for any analysis. Now, We have done research, extensive research on how intensive samples could be translated into simple samples so that producers and those who advise them could use that. And for that, we actually use permanent, semi-permanent rings like this spread on the land. We count the insects and so on, trying to establish whether or not they have a random distribution, a clump distribution, or a regular distribution, for example, in the simplest possible case. So when you see sampling recommendations online, it's usually because they started out at a much more detailed research level and they've taken the essence of that sampling down to the minimum that would be efficient and useful for you. These are rings we use. I use, this is actually brake line from trucks, works quite well. In Africa, I use discarded medical tubing. But anything light and simple, you could throw it out there and count. But you really could just put a stick, a yardstick on the end of another stick and do quick counts and eyeball as you go. Now, of course, people know the problem that insects are spread through the field. You might have a small number spread randomly. You might have a larger number in one corner. So the solution for that is to do stratified sampling. So the field is divided up into maybe 4 sections or 6 if you're keen on getting the best estimate. That's an important attribute. Now, sometimes I've sampled under conditions where it almost didn't matter. The numbers were so high, like locusts in Africa, that getting an estimate and a variance was not important. What was important was geographic variation. Field to field, for example, in this case, these are sorghum fields in Africa. We have much better starting situation here with our fields laid out a little more regularly, but also geographic. So I'll talk about that a little bit later here. I just want to point out that the same sampling methodologies is used to refine dose targeting of insecticides in control experiments, for example. So those of you who've had the pleasure of going out and standing at an airplane and measuring the distribution of particles, for example, spray particles, have used these water sensitive cards before. You can see a beautiful pattern there coming out of that high volume sprayer. Of course, it's a beautiful pattern. In this case, we're spreading the spores that can control insects for a future biocontrol agent. But we wanted to make sure we were getting them spread out and hitting the insect and not blotchy. So the same sampling methodologies, calculation of mean, variance, standard deviation, standard error, confidence intervals, are used at the research level, but they could also be used fairly quickly and simply at the management level if we're looking at the distribution of pests. Okay, once we have those numbers, I won't dwell on this, but I'll have one slide here. We sometimes draw conclusions rather quickly, and we have to be careful because the life cycle, the phenology, the number of natural enemies present, and the feeding dietary preferences, for example, of the pest, these things all matter a great deal. I mean, a classic example is grasshoppers. There are certain species that will not feed on broadleaf plants at all. So we can't just count grasshoppers, we have to identify them. And there are others that won't feed on grasses, oddly enough. So if we go out and we find some damage and we look at some counts, we have to be careful before we determine that it comes from that particular pest on that particular day. So there's two kinds of errors regarding correlation, assuming that a correlation, just happening together indicates that a cause. And the other is ignoring correlation because you're worried about that first problem. The second one is becoming more and more common because everyone's repeating number one like it's a mantra. There are common problems with validation. If we have a model or an understanding for what a pest does in the field and then we make a prediction and we find out that it comes true, we sometimes think the model is correct, and that's not necessarily the case. And sometimes we actually have reality that we don't want to accept because we don't like the answer. So there are all kinds of interpretation issues after the sampling comes into play. But the starting point is the sampling. Now, some of the sampling is done by a producer or those who advise them. Others are done by field personnel. And as you may know, every year for a century almost. Actually, more than a century, but in different levels. Field personnel have gone out, they call themselves the Alberta Agriculture Fieldmen, in our case, and do the grasshopper survey. And they normally do it in the late summer. Now, I wish they did a spring one because it's very important, but it's very intensive work. You can see here that in this case, it's obvious what's happening, but what might not be obvious is that these are all the two-striped grasshopper. So they present a particular risk. So insect species matters, numbers matters, and geography matters. And sometimes the threshold, like I said at the beginning, has to be taken with a grain of salt. Here's one I saw June 14. And I'm not really in the business of sampling grasshoppers and looking for damage and all that anymore, but I have to admit it's in my blood, and so I do it when I can, and I do it even on the weekend all over southern Alberta within driving distance. And I mostly have been finding very low numbers this year. But here I found 100 per square meter. Now the threshold is about 12, right? So 100 got my attention. I swept, and 3 or 4 swings of the net gave me those grass. 7 or 8 times the threshold and no damage. It's hard sometimes to see what's going on with insects. And that's why sometimes you have to make a decision to come back and sample later. Because with grasshoppers, the number is so highly variable. If we say there's a threshold of 12 or 15 or 2 or 10, in a way, it's just a feeling, because the spread, the range around that threshold is so extreme that it hardly matters. I've seen 40 cause no damage. I've seen 8 cause extensive damage. And it has to do with the temperature, the species of grasshopper, conditions, how windy it is, and so on. So with grasshoppers, fortunately, they don't spread a disease. They don't do anything like that. They don't suck the juice out of the plant. They just chew. So look for damage. You can save yourself a lot of trouble by estimating grasshopper numbers very broadly in general. I saw a lot or a little. I saw more than 20 per square meter or way less. And damage, look for damage. Damage is the most important thing. So sometimes the counting is not the main thing to do. Sometimes it's to come back and check again, check another edge of the field. I don't know, I just thought I'd show this photo because even when I brush them away, they're still sitting on the leaf. And you notice that there's not a single bit of chewing damage on that crop. So it's a mystery to me somehow, even after all these years, how sometimes the counts really have to be taken with a grain of salt. Stay skeptical. And by the way, skeptical doesn't mean disbelieving everything. Of course, it means don't believe too quickly. Wait until you have evidence. Evidence is everything. And I thought I'd mention, since I just mentioned grasshoppers, these are the important ones for us in Alberta. Two-striped on the left, top middle and the lesser migratory, which is almost absent recently. It was very, very big in the 30s, the 40s, the 80s, and some other years, but lately it's minimal numbers in most places. Packards, clear-winged, is a species that shoots up to large numbers, feeds on wheat, barley, and grass. That's the one that caused the enormous grasshopper outbreak in the Youngstown, Consort, Coronation area. 2001, they were growing, and I put out a news item on it. 2002, 2003, they were flying everywhere. Here's the interesting one. Brunner's is now up in the Peace River. It kind of came out of the trees in a way. In the 90s, I noticed it eating pine trees even, and wiping out alfalfa, and predicted that it would become a big pest. Well, it didn't become such a big pest down here in the south or central, but it did in Peace River. So this is a survey data from the fieldmen, and they deserve a huge medal, in my opinion, for doing this year after year. And we put that into GIS analysis ever since 85, I guess, was the first one, and produce a map that looks like this. This is the Alberta Grasshopper Survey. And it's smooth together, and there's some missing spots, and there's some holes, and there's some estimation. So really all it does is say whether or not you should be paying attention in June or not. It doesn't mean that there'll be that many, because those numbers are the breeding population. I mean, for example, there were a few years ago, there was a study that said lentils at a low threshold in some cases with some species of grasshoppers of only 2, because they can clip the pods. But that 2 has nothing to do with this number here. This is the breeding population. 2 in the spring, you could easily get from any of these numbers. That's just the old map, goes way back. This is 1989. We thought it was pretty fantastic. You could make these on a printer back then. Okay, species determination is very important. This is the clear-winged grasshopper at the top. This is the Brunners at the bottom. Those are both big pests in the Peace River region. Brunners is easy to recognize because it's so dark. This is a 5th instar, this very dark grasshopper. And when I was up in the Peace, not last year because of the pandemic, we didn't do it, but the year before I went up and found many cases in which this grasshopper was in large numbers along the field margins, eating the thistle and not eating the wheat, the cereal crops at all. But alfalfa, canola, a few others, it will certainly do so. Now, life cycle, I wanted to mention, we have to consider that, of course, with insect pests. Fortunately, they're straightforward. There's only 2 kinds. The grasshopper kind hatches from an egg, looks like a grasshopper, goes through stages, about 5, and finally gets wings and then lays eggs again, 1 generation per year. The other life cycle, of course, is have an egg, larva, pupa, adult, like beetles and moths and butterflies and so on. But you have to know what's in the field, and you can ask somebody or you can continually check. Another thing that matters is development rate. Now, since about 1910 or something, they've calculated degree days and used it in orchards and even in prairie agriculture a bit. It's just a method of trying to estimate how hot it's been and whether or not there's been enough heat accumulation for insects to hatch and grow and do their thing. Well, we've actually made a little model that fits that line better, this curve model. But fortunately for us, it's fairly straight in the zone that we're normally in, between 20 and 35 degrees. And we know that curve for the different species. So the ones that have a steep slope will hatch more quickly when it starts to warm up. Those that have a shallow slope will hatch a little later. And this one is hatching now late. This is a clear-winged, little black and white grasshopper. Now you have a mixed age class, but like I said, never fear, it doesn't matter. The age class is important to people who really are trying to discern what the particular insect is doing. But the thing that's important to a producer is whether there's damage or not. Grasshoppers have that one advantage, that they signal whether they're a problem in advance by starting damage that's highly visible. Now, the last thing I want to get on to here is monitoring natural enemies. We don't do much of it here. For 20 or 30 years, we've recommended it, and we've done research on it, but it hasn't been a formalized survey very often until this group that's doing the field heroes thing you saw a minute ago. That's nice. They're working on at least drawing attention to them. This ladybird beetle is eating an aphid. It's an example of a natural enemy. In places like New Zealand, they actually count the pests, and then they actually count the natural enemies as well, and then do a kind of a ratio calculation and use that as their threshold. We could do that here. We could do it especially in crops like potatoes. In fact, I think it's a little bit inadvertent because we haven't had much Colorado potato beetle in a long time, but the potato fields have been using so little insecticide spray that a complex and very healthy community of natural enemies have built up in potato fields. And even with crop rotation, they find those potato fields and they are there essentially guarding those potato fields. So they're feeding on thrips, they're feeding on aphids and so on. So if a new pest like the potato settle I mentioned comes along and lands in that field, it probably will be eaten, and that's good. But if we get back into a cycle of using a lot of insecticides, if we must, for example, with maybe a Colorado potato beetle outbreak in the future, we won't have that buffer of natural enemies there. And if potato cells come in, for example, they could establish. So it's worth counting them, but it's not easy because there's so many different kinds. Of course, there's rove beetles, there's spiders, and spiders, by the way, do a lot of economic value for us in terms of removing pests. They're feeding on ligas and all sorts of things left, right, and center, but tiny natural enemies as well. These mites attack grasshoppers. These maggots attack grasshoppers. We used to think it was a curiosity, 2 or 3%, 5%. But last summer, I found that the clear-winged grasshopper had 50% infestation with maggots. That is extremely high. A fly comes out. I mean, the maggot comes out of a grasshopper and forms a pupa, and a fly comes out. So, when we say we want to reduce insecticide use, it's not just for the sake of general environmental protection or saving money. It's also to protect some of these natural enemies that are a little bit too complicated to find easily and know about, because we don't have many people who are specialists and out there looking. It would be like if we had like 3 medical doctors for Alberta and asked them to cure all the diseases there are. So these are the pupae from natural enemies that come out of grasshoppers. They can easily be surveyed. I survey them as a hobby every year, and I see fluctuations up and down. And sometimes it looks like an outbreak that's been going for 2 or 3 years builds up a lot of these natural enemies, and it helps to bring it down, and that's worth money. Okay, that's all the photos I have, but I thought we could leave some time for a little discussion. Let's see if I stop sharing. Am I visible again, Jamie?
Yes, I see you.
Sure. Okay. So I'm not an expert in all insects or anything, but I do know something about the numbers and the life cycles and the biodiversity. And if there's any questions, I could either answer or tell you who to see.
Okay, so there is an opportunity to ask questions. You guys can type it in the chat or raise your hand.
I better open the chat. Can I?
The conversation bubble.
Oh, show conversation. There it is. I've been using Zoom all year, every day, but not Microsoft Teams. Oh, no, sorry. I put my hand up to wave goodbye to Scott Gillespie, and then I forgot to take it down until after lunch.
Not a problem. Tim has a question for you, and he's saying, how likely are we in Canada to adopt New Zealand model regarding natural enemies?
That's a good question for the provincial government agencies, and I know that they do care about this, and I know that when we wrote our potato cell recommendation at the end of 2017, that was one thing we did recommend, was monitoring those natural enemies. And we're fortunate in that although there are a lot of species in each group, for example, we found almost 20 species of ladybugs in potatoes. They, as a group, do one thing basically. So we're fortunate that we don't need to monitor all species, but the problem is some of them, of course, are very small, like the Hymenopterus parasitoides, the little wasps that attack aphids. The canola people are paying a lot of attention to that sort of thing, and I have a feeling that if monitoring of natural enemies becomes a regular feature that really pays off, it would either be in canola first or possibly potatoes. People like Scott or others might know.
Okay, so we have Brian Kennedy, your hand is up. Do you have a question for Dan?
I do, thank you. Dan, looking at the forecast maps and the weather forecast of really hot, dry weather across Alberta for the next 7 or 8 days, how can farmers kind of interpret that on whether they should take action sooner than later, or increase their scouting, or anything that they can do looking at those two forecasts?
I think your second option is the better, increasing scouting. Now, cabbage seed pod weevil seemed to be low for some reason, and I didn't know why. I mean, I'm not looking for them specifically so much. I did 10 fields only. But I used to see a lot more turning up in the net when I'm doing roadsides, and I have a feeling that they were slow to get going, and grasshoppers were oddly slow this year, a little bit late. Now what the hot weather will do will totally catch them up. That lateness will be nothing but history now, and they'll be caught up to third, fourth, fifth instar, and moving. So I wouldn't start spraying necessarily if you don't have to, but I would do a little more scouting, because they've been hiding in a way, not really hiding, but hidden in roadsides and not really on rangeland, but in certain pastures because of their small size and their lateness. Like, for example, the clear wing grasshopper, little black and white one I mentioned, it only was hatching last week. That's 2 weeks later than some years. So they've been kind of hiding in there, and now with this heat, they will blast up they'll totally catch up. There are years in the past in which, I think it was 2018, we started out very slow and then totally caught up. Many of the insects did. So I would say a little more scouting, a little more being aware of damage, a little more use of the net. I wouldn't overdo it. It's maybe not worth it, but at the end of 3 or 4 hot days, a lot of these insects that were behind will suddenly be noticeable. And like you say, a good starting point is looking at that forecast map, all of them.
All right, fantastic. Scott, you have a question as well for Dan?
Okay, now you can hear me? Okay. Actually, I think Dawn pretty well asked my question, was about these insect refuge areas. And I guess what I was going to ask is whether we need to worry about the pollinator blends that people are selling, or do we just encourage natural areas to, like maybe unproductive areas of fields or margins, just let them go natural, or do we need to try to get in big pollinator strips in fields?
That's a good question, and I don't have an answer for that, but I'd like to see more research on that. I keep hearing plans for that work, and I think that's fantastic. No, I think that's really good, and I'm really encouraged to see more work with bees at places like Agriculture Canada. I was afraid some of that would be curtailed with slowdown in research over the last decade, but it's up and running and it sounds good. By the way, could I mention one thing that relates to the last question, and might actually relate to the pollinators a bit too, and that is, I'm often asked about winter conditions and whether or not that will control or reduce the pest populations. And I just want to point out that that's very rare that happens. If you put a thermistor, thermocouple, what have you, some temperature measuring device in the soil, you'll find out in a hurry that the insects are fine. If it's minus 25 in the air, in the soil it's typically minus 9. And it takes minus 15 before they begin to drop off. They have super cooled conditions based on cryoprotectants like glycerol and different sugars as well, other alcohols, but also sugars. And minus 10, they don't die. So if it's minus 25, it probably hasn't even hit minus 10 where they're sitting, because they snug in an inch or 2 or 3 inches deep. So before that question comes up about the cold, I don't think it's a big factor ever. Maybe once I've seen it, but in that year it killed the winter wheat as well. So.
Okay, I have a question from a student, and he's asking, how can I improve scouting on field when looking for insects? Tips, please.
Well, there's a few things I would recommend based on experience with a lot of scouting that goes good or goes bad. One is don't underestimate how weather dependent the insects are. It's a funny thing. We did a spring survey one year, a very extensive one paid for by the province of Alberta back in 2002 or so. And we did one in the 80s as well. And I did a huge multiple regression to find out what factors determine the grasshopper number. You know, is it the vegetation? Is it the date? Is it the species? And one of the biggest factors of all was the time of day that they sampled. So I would say don't underestimate the impact of weather and the time of day on insects. Get to know the insect behavior, maybe even repeat sample. Another one is, I would say, don't consolidate samples. I know it's popular for people to do that. They'll, if they want to do 4 sets of 10 sweeps, they'll just throw them all together in one bag. But most of those could be from the last couple sweeps. You know, I would say do get an estimate of the variability. Do calculate that, even if it's just the range. And if anybody wants to, because of a personal interest or curiosity, see examples of how you can calculate what proportion of that field might be above the threshold and below the threshold based on the variability, I could send you some files or build them on the screen for you. But I do think a little more appreciation for variances and precisions in the field will help us. Partly because some insects are just predictable, dead steady. Others are all over the map, and we need to treat them differently. One thing with that sweep net, by the way, if you want a practical suggestion, pardon my voice, I'm a little hoarse today. If you want a practical suggestion, compare the sweeping results from multiple people. We have huge variability, and the reason we do is that not everybody sweeps the same. If somebody tells you one number and someone else tells you another number, you really need to know, were they sweeping 180 degrees? Or were they just dipping it like a, like a water dipper into the insects? Or what exactly were they doing? Were they moving quickly? Were they letting them get away? We can walk down a field with 3 people and get 3 different results. So it does take a little time. I guess that would be my recommendation. Since you use the word student, it would mean you don't have years and years of doing it. So calibrate yourself. The calibration takes time, and I can't recommend it to everybody, but it is a fun thing to do. At the Grasshopper Survey Field School years ago, I used to cut up paper of different colors, about 1/4 inch by 1/2 inch, so grasshopper size, sprinkle them all over a hayfield, and then we would go out there and we would count them and see how many we thought there were and what proportion were green, yellow, or white. And it's really quite interesting to find out how you have to take more samples, take a little more care before you've really calibrated your own field counting. The last thing I'll mention is be aware of species, be aware of their biology. It's not wasted at all to know all about them. You could even go to iNaturalist and things like that and look at the different species and look at where they occur in Canada and the US, the timing, the color forms, just get to know them. So when you see them, you're not always scratching your head. What is this? Is this a pea-leaf weevil? Or is this an alfalfa weevil, or what? Is this a 2-striped grasshopper, or is this a red-legged grasshopper? It's really important, I think, to get that biology, that lore down. You almost end up being as fanatical as the wildflower people. But in the end, it's worth it.
I know some people's body mechanics are different than others. Dan, do you have any comment on doing the 180 sweep versus just a 90?
It's a tough one because it depends also on how fast you walk. This sounds a little funny and eccentric and embarrassing, but years ago, my son, who's a geophysicist, was listening to me complain about how our sweep samples weren't working out because everyone sweeps differently. And he said, oh, I can calculate that. It's a simple moving torus. So he actually made a mathematical model with differential equations and geometry as well on how that circle swings ahead of you and then comes to the end and flattens and then swings ahead of you again as you move forward. And we modeled it, and we found that the 2 most important things by far were how quick you walk And if you go all the way to the side, or if you raise it up, then you're not really sweeping a full sweep. So it's quite simple for me to go into a field and say, hey, I got 9 cabbage seed pod weevils for 10 sweeps. And someone else say, well, I got 1. And it's because of this dipping the net up and down. It should be flat with the landscape and hit the vegetation. Yeah, it's funny, it takes time. It's kind of weird to study mathematics and physics and statistics, and then you find out that your biggest problem is you can't hold on to the net properly. That's why I put nails on the handle. It doesn't spin in your hand. But if you're only doing 30 sweeps, don't worry. I'm doing 1,000 a day.
All right. Does anyone else have any last questions for Dan before we wrap up this presentation? Don't be shy.
Okay, maybe I'll just on that topic of the sweep netting. Can you do twice as many 90-degree sweeps as 180? Because I have that problem that when I'm twisting my body too much, I eventually hurt my back. So
it's true. I know that's absolutely right, Scott, and it happens to me. And maybe you noticed, if it showed clearly enough, when I was sweeping, I wasn't really doing a 180. I was doing a 180 plus, about 20 on each side. And the reason I was is I know that those first ones hardly count because they're too slow and they're not done properly. So I was kind of compensating. So you could compensate the other direction too. So if you just do 90, the problem is that's the point of where you're walking. So if you do the first 90 or the second 90 or the middle 90, those things matter. So I would say, if you ever do find a decent, and by that I mean high density insect population, you could calibrate yourself right there. You could just say, okay, I'm going to do some 180s, do some 90s, and now I know that I'm running 3/4, you know. And I think if each person calibrated themselves, that's the way to go. We actually did try to make it standard for all people before, and it's a tough one. And then maybe we get a molecular biologist or somebody out there who doesn't know about these things at all, and they just go like this with the net in the middle and come back with 0. So.
All right. Well, thank you, Dan, for your presentation. I thoroughly enjoyed it. I think everyone did as well. I will put the QR code up for those of you that are getting your CCA credits, and you can scan it for this session.